Well, thank you very much, David. And first of all, let me thank the Society for this wonderful honour. It really is a huge pleasure to be here. I was taught years ago never to start a talk with an apology, but I ought to warn you that you might experience the smallest twinge of déjà vu as we go through this talk, because I'm going to be mentioning thyroid hormones, and you're probably sick to death of thyroid hormones already. But I want to show you a new role that these hormones play in a really important timing system that controls annual cycles. So let's start the story by going up to the high Arctic and to this island here, island group Svalbard, which means cold coast. And this island here contains the most northerly population of mammals on the planet, about 800 miles from the North Pole. And these are reindeer that have actually got here by walking across the sea ice at some stage about 10,000 years ago, around about 1,200-mile journey. So it's quite an incredible feat to have pulled this off. And they've been there for about 10,000 years, and they're resident on this island, and they're one of the most seasonal mammals on the planet. This shows the distribution of conceptions and births, normalized to 0, and 90% of the births in this population occur over a 10-day period. And these animals have to cope with a very extreme climate. The growing season is no more than a couple of months long. The only other resident mammals are actually polar bears, which live on the ice, but they're not smart enough to catch these deer. They're not a real predator for these animals. And it turns out that animals in the very high Arctic have got very deep-rooted clocks that drive their biology and their metabolism and their reproduction. But that same system is actually almost a universal mechanism in nature and occurs also in tropical animals like elephants. So what I want to do today is to tell you a little bit about timing mechanisms in general. And in particular, I want to focus on a really important question, which is how the daily clock is used to drive a seasonal cycle. There's overwhelming evidence that that is the case. Seasonal organisms won't respond to environmental stimuli unless they're presented in modulos of 24 hours. So the internal circadian clockwork is unambiguously involved in measuring time over a course of a whole year. So that's the daily clockwork, which Mitch Lazar gave us such a beautiful talk about earlier on today. And I want to talk today about another time scale which operates over a period of 12 months, which drives multiple aspects of physiology in wild and domesticated animals. And the man who really drew our attention to the importance of this system was a German plant scientist called Erwin Bunning, who was immensely distinguished and a foreign member of the National Academy of the USA. And nearly 80 years ago, he proposed a really elegant model which explained in his mind how the circadian clock might be used to drive an annual timing mechanism. And he christened this the external coincidence hypothesis. And I'm not going to burn 5 minutes of my time in telling you how this external coincidence model works, because you'll see very clearly what it is when I run through the story I'm about to tell you, and we'll come back on the last slide to this man, and we'll look at how this idea that was proposed 80 years ago has only just been tested in the last few years. So just to drive the point home that seasonal cycles are both fascinating and important, this is an example of a widespread laboratory animal that's now used by colleagues of mine and myself in the past. That's the Siberian hamster, Phodopus sangorus. And it's a highly photoperiodic seasonal species. And if you take these animals and you put them in natural photoperiods from the middle of the summer and then just track their body weight, you'll see that they will spontaneously lose body weight. They're kept on a constant available food supply throughout. And they'll lose around about 40 to 45% of their body mass, losing virtually all major fat reserves by the time they get down to the middle of the winter. And if you food restrict the animal at this point here, the animal will accelerate its body weight loss, so it's on 70% of its ad lib. But if you de-restrict it at this point, it will bounce back up, but only to the point that it would have been on if you'd left it alone in the first place. So it knew where it wanted to be. There's an internal clock driving its biology. You can do it again down here, and it bounces back up. This is a photoperiodic process because if you keep the animals on constant summer day lengths, they just bounce back up to the high level that they were on before. And here we've got an example of the species, and this animal here, one individual, about 400 photographs over the course of a whole annual cycle, and it's now in the winter state, okay? It's now lost 40% of its body weight. The testes are shriveled up. It's now lost sensitivity to leptin. Prolactin levels are coming back up. Its testes are regrowing. It's laying down fat reserves. It's gaining around about 40% back of its body weight. And it's back there 12 months later to where it started. So that's the seasonal clock in 28 seconds in one very cooperative hamster called Herman. Well, I used to work on hamsters, but I quit working on these animals for several reasons. First of all, the brains aren't big enough for the kinds of very detailed anatomy that we'd like to do. And secondly, I was very attracted to working on these animal sheep because they're still robustly seasonal, but they've got a beautifully defined reproductive physiology thanks to a marvelous history of research in this country, France, and the USA, and elsewhere. They're ideal for anatomical studies, but most importantly of all, is now turning out to be a real asset. This is the first and so far the only mammal that we have in which we have a fully sequenced annotated genome which retains all of the seasonal, robust physiological responses that we want to study. Now, you probably all know that the main hormonal signal that drives seasonal biology in mammals is the hormone melatonin. And melatonin is produced from the pineal gland, shown here. That's in a rat. That's a scanning EM. It's heavily vascularized tissue. And here it is in man. This is one of the hemispheres removed, and that's the point of location there. And the pineal gland is linked by a multisynaptic pathway involving ganglion in the neck through to the paraventricular nucleus, and then back to this structure, the suprachiasmatic nucleus, which is a ventral hypothalamic structure, which is charged with the main role of keeping the body in time to the external light-dark cycle. And the main product of the pineal gland is this hormone here, an indolamine, melatonin, the structure of which was described by Aaron Lerner following isolation of 250 kilograms of cattle pineal glands in those heroic days of early endocrinology. And the hormone that it produces is sculpted to the night length. So it provides the animal with a biochemical representation internally of what the external photoperiod is, because it comes up at night and goes down in the day. So that on winter day length, the duration of the signal is long, and on summer day length, the duration of the signal is short. Now, this whole mechanism generating this rhythm here is driven by a clock in the suprachiasmatic nucleus, and that's a piece of suprachiasmatic nuclei, two suprachiasmatic nuclei in culture. That's a fusion protein for luciferase tagged to one of the clock genes there from one of our transgenic vermin that we keep at Manchester. And this is the circadian cycle that actually drives this oscillation in melatonin. Well, with the discovery of melatonin and the recognition of its prime importance in driving seasonal rhythms in mammals, you'd think it would be a pretty quick business to track down where it was working. And I got very involved in this over many years using first radioligand binding and then later in situ hybridization studies to try and identify obvious target sites in the hypothalamus. And it turned out to be a complete veil of tears because none of us could find consistent structures where melatonin bound in the hypothalamus. And the penny dropped when Peter Morgan at the Rowett Research Institute in Aberdeen published a series of quite beautiful papers showing that the major site of action of melatonin was likely to be in the stalk region of the pituitary gland called the pars tuberalis. And there it is there. That's a dissection from the ventral brain region of a sheep. That's the optic chiasm there. That's the pars tuberalis, and that's a bit of the main pituitary body there that's being cut off. And melatonin acts on a G-protein coupled receptor. In fact, there are several, but it acts on just one of them, MT1, to encode the seasonal signal. And it's this structure here that I want to spend most of the rest of the talk telling you about. And I'd also like to remind you that if you ever remove a pituitary gland from an animal, that's the bit you break off. So it's very, very poorly studied, apart from a small group of us who are interested in seasonal timing. Well, it turns out that the pars tuberalis has got a unique population of hormone-secreting cells in it. And these are thyrotropes producing TSH. But these cells come from a different embryological lineage. There's nice data here from HES1 knockout mice. They also lack the receptors for TRH, and they don't respond to TRH. So the TRH signals, which come into this tissue from the brain, bypass these cells and act on the main thyrotrope populations in the main pituitary gland here. So you've got a rather odd group of cells up here, which we know co-express the melatonin receptor, but don't have the capacity to respond to TRH. And they're anatomically very closely associated with a group of ependymal tenocytes, which come from the ventral hypothalamus, which I'll tell you a little bit more about in the next slide. So here they are. These are the tenocytes. This is a coronal diagram of the ventral hypothalamus, the median eminence down here, and this is the pituitary body at the bottom here. And these tenocytes are one of the main thyroid hormone-converting cells in the brain, the ventral hypothalamus, and many of them project down into the pituitary and have physical connections to the cells of the pars tuberalis, and in fact to those TSH-expressing cells in the pars tuberalis. And it's in this region of the brain that we find strongly expressed enzymes that are involved in the regulation of thyroid metabolism, including, as we heard earlier, and from Teo Visser as well, monocarboxylase transporters, that's MCT8. And you can see that beautifully maps onto the tenocyte population in the ventral hypothalamus. So we've now got two bits of the story. We've got an unusual TSH population, and we've got a very close apposition to thyroid-converting cells just due north. Well, a series of studies have been done now, both in birds and in sheep and in hamsters, showing that the beta-TSH subunit of TSH is strongly regulated by day length. And this shows some coronal cross-sections, in situ cross-sections of the pars tuberalis. It's always horseshoe-shaped, by the way, because when you take a brain and turn it upside down, the pituitary sort of flops over the curved surface. You then freeze it, and you end up with a horseshoe-like structure. In fact, that's what you're seeing here. And you can see the melatonin receptors are localized explicitly just to this region here. And here's the beta-TSH unit, which is only expressed on long day lengths, but is virtually absent on short day lengths. Now, beta-TSH is also expressed, of course, in the pars distalis, the main pituitary body, and there it doesn't change with photoperiod. So there's a timing mechanism regulating beta-TSH expression in just these TSH cells in the pars tuberalis. The alpha unit, by the way, the subunit, doesn't change with season. A little bit more about that in a minute. So the summary so far is that the pars tuberalis expresses melatonin receptors and contains a unique population of thyrotropes, but they don't respond to TRH, and the beta-TSH subunits under photoperiodic control, and it's activated on long days. So the question now is, what on earth is this beta-TSH population doing, and what role might it play in seasonal timing? And the answer seems to be that the TSH of PT origin is actually driving thyroid hormone metabolism in a seasonal manner in the ventral hypothalamus. The TSH receptors are expressed in the ependymal cell there. You can see that over there, and that's the beta-TSH activation on long days there. And if we take this tissue here and culture it in the presence of TSH, we can elicit cyclic AMP release here, shown in this line here. But if you co-culture it in the presence of melatonin, you suppress cyclic AMP activation by that structure. And here's the rise in the beta-TSH over time. That's day 1 of long days, and there's day 10. You can see it's right up. And associated with that, we see strong activation of deiodinase 2 enzyme, which, as you now all know, is involved in the conversion of T4 to T3. And by day 10, levels are very much higher. And here's the in situ hybridization for DIO2. You get a very, very weak expression here on short days, but if you put the animals onto long photoperiods, you get very, very strong expression here, particularly in this ventral hypothalamic region adjacent to or part of the tanocyte cell population. So it looks like TSH of PT origin is activating thyroid hormone metabolism in the adjacent hypothalamus. And here's what we think is going on. On long photoperiods, we have a drive towards the production of DIO2, leading to outer ring deiodination and acting on nuclear hypothalamic receptor targets and activation of T3. And on short photoperiods, and I don't have the time to go through the story today, the other system switches on, and DIO3 becomes the dominant enzyme. And that, of course, drives inner ring deiodination and then via DIO2 to T2, which is biologically inactive. So the long-day signal is activating the active form of thyroid hormone within this local population in the hypothalamus. And here's a little cartoon or model for you as to what we think is going on. Here's the ependymal there with the cell body of the tanocyte projecting down to the pars tuberalis. And here are the PT thyrotropes, which are organized quite interestingly in structures with cisterns around them. And on long days, TSH is activated, a short duration melatonin signal, driving up DIO2, increasing local T3 concentration, and leading to remodeling of structures in the hypothalamus, and as we now suspect very strongly also in the pars tuberalis. Whereas on short days, this process is switched down, DIO3 comes up, DIO2 drops down, and the system goes into reverse. So we've now got a thyroid switch mechanism which is activated in a photoperiodic manner dependent upon the regulation of TSH. There's quite a lot of evidence now that T3 is indeed the signal, because if you take a T3 implant and you stick it into the ventral hypothalamus of a seasonal mammal, you can mimic the effect of putting it onto long summer day lengths. So T3 is read as a day length, long day signal, and it will activate the reproductive system as if the animal had been put onto summer day lengths. We don't yet know quite what circuits the T3 system is engaged with within the hypothalamus, but evidence is now emerging, particularly from lovely work by Valérie Simoneau in Strasbourg in France, that the RF-amide neurons in the brain are prime targets for the action of T3 in activating this reproductive response in a seasonal manner. So the most parsimonious model that we have is that T3 acts on RF-amide neurons. And I could have put a very similar cartoon up showing similar possibilities for melanocortin signaling and changes in activity of that axis, which we also know is under photoperiodic control. So the summary so far then is that TSH acts on a receptor field in the adjacent tanocytes and drives deiodinase expression to regulate local T3 availability. So now we've got to work out how does the PT, the pars tuberalis, tell the time, and what is the switch that drives TSH beta? So to address this, we went back to basics and started doing deep sequencing and arrays, and thank God we had a sequenced sheep genome. And we were able to identify following photoperiodic exposure, this is the 7th day of long day lengths when sheep are put into artificial photoperiods, we managed to identify a rather curious gene called IR3. This is upregulation from left to right, and these are things that come up in the summer, and these are things that come up, these are things that are downregulated in the summer. And there's around about 15,000 genes on that plot. And this is a log scale, and that gene there is around about 100-fold higher than the baseline. So it's an absolutely massive signal, and it gets even higher by the time you get out to day 28, a month later, is way up there beyond the reach of all of the other genes that are expressed in this tissue. When we do the in situ hybridization, we find that IR3 is exclusively expressed in the pars tuberalis, and it's strongly activated by photoperiod. In fact, it comes up on the first long day that you put the sheep into. It's the first signal that the sheep can read, and it reads it within about 12 hours of seeing that long day signal. Now, the IR genes are transcriptional cofactors, and you heard a lovely story about this from Bert O'Malley yesterday. And these genes are involved in retinal development, or thought previously to be involved in retinal development. There are 4 of them in the mammalian lineages. 2 of them, when they're knocked out, are lethal, and 1 of them, when it's knocked out, leads to a pretty sick mouse. But the IR3 knockout is mild and virtually normal by comparison to the other forms of this gene. And in Drosophila, if you overexpress this using GAL4 drivers in different tissues, you can induce full-blown retinal development in the wrong part of the body. So what on earth is a retinal determining gene doing involved in seasonal timing? So the first thing that we did was to raise an antibody to IR3. I think there's only one in the world, and it's raised against sheep, IR3. And on short photoperiods, in the pars tuberalis, the only protein that you can reliably see there is the alpha subunit for TSH, that's GSU there, shown in red. But if you put the animal onto long days, long photoperiods, IR3 can be detected as a nuclear antigen, and you can see also expression of beta TSH, which is the green signal here, and beta TSH and IR3 very strongly co-localize. And that's shown in a bit more detail over here, where TSH beta is now green in color and IR3 is red, and you can see that around about 75% of the TSH-expressing cells also contain IR3. But if you put the sheep back into winter day lengths for 4 to 8 weeks, all that signal goes away. You can't see anything at all. And in the pars distalis, of course, TSH beta doesn't change at all. And Hugh Dardenti, working then at the University of Aberdeen with Dave Hazelrig in a collaborative study with us, mapped out the profile, it's quite a heroic study, using in situ hybridization on winter day lengths, and then the 3rd day of summer day length, the 15th day of summer day length. And you can see that the TSH beta signal is pretty flat in the winter, and it comes up by day 3, and by day 15, that's the magenta signal there, it's way up. And here's the IR3 signal, and you can see that on winter day length there is a little tiny oscillation. It comes up about 12 hours after the onset of darkness. I'm going to come back to that point right at the end. But by the time we get to day 3, the signal's way up, and again by day 15, way up again. So the obvious question now is, does IR3 regulate TSH beta? And it turns out that the TSH beta promoter has a conserved D-box in it. Now, D-boxes to circadian biologists indicate that you've got something interesting there, because D-boxes are acted on by a class of PARZIP proteins, of which, including TEF, HLF, and DBP, the biology of which was first worked out by a Swiss colleague of mine, Uli Schibler. And this conserved D-box occurs right throughout the mammalian lineage, so it looked interesting. Furthermore, one of the D-box proteins, TEF, thyroid elongation factor, actually binds to a synthetic oligo for TSH itself. But when we looked at the temporal pattern of these PARZIP proteins, none of them showed any obvious change with photoperiod or even with time of day. So it looked like they might be necessary, but it looked like that something else might be playing a role in this story as well. And the answer to this came when we co-activated and looked at the action of the co-activator of IR3 on TEF action on TSH. And this shows the story here. Here's TEF, and here's 6-1. It's one of the other proteins involved. And then if you increase the amount of IR3 plasmid, you can dramatically increase the expression of a beta-TSH promoter construct. Now, as Bert O'Malley reminded us yesterday, co-activators have always got enzymic activity as well as co-transcriptional capacity. And this particular co-activator is a phosphatase. So we mutated the phosphatase domain of IR3, and it didn't change the activity of the gene whatsoever. So here's the story. On short day lengths, IR3 is switched off. 6, TEF, and DBP are around, but the beta-TSH promoter is very weakly activated. And on long days, this signal comes shooting up, and it co-activates TEF, possibly DBP, and 6 proteins, driving TSH beta expression. So that's how a co-activator in the PT drives the seasonal response. But I haven't yet addressed the really central question, which is how the circadian clock locks into this process. And that's the last 3 or 4 slides. And that's the question. How does the circadian clock work in the PT operate to drive seasonal responses? Now, there's a little bit of biology I need to explain to you before we go to the last couple of slides. And that's about the biology of melatonin action. We've known for a very long time that melatonin inhibits cyclic AMP signaling, signals largely through GI. And so when melatonin is present, cyclic AMP is repressed. And when melatonin levels drop at the beginning of the day, you get a de-inhibition and cyclic AMP activation. We've also known for quite a long time that in the pars tuberalis, there's a local clock gene rhythm that's driven by the melatonin signal. And at the onset of night, cryptochrome, which is one of the key transcriptional repressors, is strongly activated following the rise in melatonin. Now that might sound like a paradox. I've just told you that melatonin represses cyclic AMP, and it does something else, which is it activates, through a pathway that we haven't yet managed to fully resolve, a transcriptional repertoire, including cryptochrome. And we think that the major upstream signal in this is another clock gene called NPAS4, which was previously only thought to be involved in memory consolidation in the hippocampus. And I just draw your attention to the fact that the hippocampus is the only other structure in the brain where we get reliable, strong melatonin receptor binding. So there's probably a link there, but it hasn't yet been resolved. So this is work by Alex West, who's a PhD in our student. So we've now got a clock gene oscillation driven by the melatonin signal. So here's what's going on. At least this is the model that we think is operating. In the brain, there's a central pacemaker operating in the SCN neuron. And at the beginning of the day, these transcriptional repressors, period and cryptochrome, are activated by light. And in antiphase to these, there's the transcriptional activators of the system, BMAL and clock. And these oscillators, through that complicated pathway, drive a rhythmic hormone signal, melatonin, which is sculpted to measure the length of the day. And there's a slave oscillator sitting in the pars tuberalis, which is driven by melatonin-induced cryptochrome 1, which then sets the phase of a local clock that runs in this peripheral tissue. And cryptochrome comes up sharply at the beginning of the night and then declines, as you can see in that in situ, towards the end of the night. So here's the model here. The central pacemaker is driving through a hormonal signal a peripheral oscillator, which then induces the downstream effect. So how does IR3 fit into this? The best model for this is that IR3 always rises about 12 hours after the onset of darkness. Do you remember those in situ profiles that I showed you? Even in the winter, you get a little peak of IR3 coming up around about 12 hours after the onset of darkness. But it can't get very far because this is a likely cyclic AMP-regulated gene, so it's suppressed. So if the melatonin signals of long duration, the circadian clock still drives the IR3 rhythm, but the rhythm can't really escape to activate on downstream targets. If we put the animal to summer day length, now the IR3 system can be activated, and it can now be read. And that is the external coincidence hypothesis that Erwin Bunning proposed all those years ago. He proposed that the light-dark cycle did 2 jobs. It drove a rhythm in a target tissue, and then the duration of the light and the dark acted to regulate the expression of clock-regulated components within that system. And it was an absolutely brilliant and insightful piece of work. And it looks now that the timing system that animals use is virtually the same as the timing system that plants use. And indeed, we've even got a little bit of evidence emerging that some of the same genes and molecules might be involved in both. So we now know that this system that I've described to you is at least 250 million years old, because my Japanese colleague, Takashi Yoshimura, has pioneered wonderful studies in birds, which have evolved somewhere around about here, in the Cretaceous, and we go right back now to the beginning of fishes, because he's also identified homologous structures in fishes. And throughout all of these organisms, the TSH receptor interacts with a photoperiodically activated TSH-beta, which in turn regulates deiodinase enzyme signaling within target tissues within the nervous system. But it's even more interesting than that, because there's a recent study that's shown that the seasonal spawning of corals may be regulated by IRE. So it may be that that transcriptional co-activator is one of the most ancient timing genes that we have. So this is a very ancient timing process, and in all of the animals that we've looked at so far, this system is persistent. So to summary, and a homage to Erwin Bunning, the PT operates as a melatonin-driven slave oscillator, and this drives the circadian IR3 coactivator, and the rhythmic expression of IR3 is maximal when it's coincident with light, and that's the external coincident timing mechanisms. Now, I've known of this literature all my working life, and it's taken me until 4 years ago to finally work out what was going on. Now, the genetic timing mechanisms that we're talking about are incredibly hardwired, and they respond to predictable cues which have evolved over very long time periods, namely the light-dark cycle. And these systems are not that plastic. And many species, particularly in the higher latitudes, may well encounter severe problems in adjusting to rapidly changing local phenology and resetting their breeding cycles. And there's evidence that that's now emerging if you look at the catastrophic effect that climate change is having on the migratory caribou populations, for instance, in North America, where you've got a clear mismatch between the time of birth and maximal food needs with the pattern of plant development and growth. So the final slide for you is, and to preempt the obvious question, what of humans? Well, we have a pars tuberalis, and we have also got melatonin receptors there. And this is the bit of the brain that tears off when you take it out, or if you take the pituitary out, it also tears off. And I'm going to set a challenge to you. I don't think I can find a single publication in our own species which has looked at the biology of this structure in relation to TSH signaling. And that's in the face of now 15, 20 years worth of growing literature of the importance of this structure in all other vertebrates on the planet. And I think it's high time that the clinical community started looking closely at this particular structure and what role local thyroid hormone signaling might play in our own biology. So to say thank you to a few people, one of my long-time standing collaborators, Julian Davis, who's here in the audience, Alex West, who teased out how cryptochrome was regulated by melatonin, very old friend and colleague, known to most of you, I suspect, Alan McNeely at Edinburgh, and somebody you probably don't recognize, apart from a few of you, that's a very good friend of mine, Dave Burt, who's a wonderful comparative genomics biologist, and he's the world authority on the history of domestication and domestication genetics. I would just say as an aside that we've now got a very strong hint that many of the genes that melatonin regulates are the very same genes that man has gone for in the domestication of birds and mammals. And that's Dave's very able assistant, Bob Payton, who also works at Roslin. Then other players shown in red, Shona Wood, who's in the audience, is doing some wonderful work on the epigenetics of this system here. Dave Hazelrig, who's now at the University of Tromsø, and Hugh Dardenti in Tours in France, and Alan and Cassia and Judith, and Helen Christian. And I don't know if you're in the audience, Helen, I'm terribly sorry, I just couldn't squeeze in the wonderful EM data that you've generated for us in this story, but that'll have to wait for another day. And Takashi Yoshimura in Nagoya in Japan. Thank you very much.